Dear students, today we are going to look at a very important topic in biology, which is cell divisions on the whiteboard. Okay, cell divisions here is divided into two types. We have two types cell divide. It's either the cell divide by mitosis or the cell divide by meiosis. Okay, we have it on the board. The cell can divide by mitosis or the cell can divide by meiosis. Okay, here in mitosis the cell divide once to produce two identical daughter cells. That is to say, the daughter cells look the same as the parents. Okay, here in meiosis the cell divide twice, okay, to produce four daughter cells that are not identical. Okay, then in cell divisions we have about 5 stages of cell divisions. These stages are the interphase, these are the 5 stages of cell divisions: the interphase, prophase, metaphase, anaphase, and telophase. Okay, the interphase stage of cell division here is the time where the cell is resting or preparing for division to take place. Okay, so nothing is happening at this stage of cell divisions, and that is why it is known as the resting stage of cell divisions. If you don't call it the resting stage, you can call it preparatory stage. The interphase stage of cell divisions, in this stage nothing is happening, the cell is not dividing, there is no change. Okay, the cell is resting or preparing for divisions. Then other stage we have here is the prophase stage, metaphase stage, anaphase stage, and telophase stage. And we have different actions, different changes in the cell at these stages of cell division. We are only going to talk on the prophase stage in this video. Okay, but before we continue with that, let's look at the meaning of meiosis. Meiosis here is a type of cell division during spermatogenesis and oogenesis. This spermatogenesis happens to be the formation of sperm cells, which takes place in the seminiferous tubules located inside the testes. Okay, and oogenesis here is the formation of egg in the ovary. Okay, and the meiosis is a type of cell division that occurs during spermatogenesis and oogenesis. That means it is the sex cells that exhibit this type of cell divisions, which is meiosis. Okay, and in this type of cell division there is reduction of chromosome to half the original number. Okay, the original number of chromosome in every human being is 46. Okay, then but during this series of cell divisions the number of chromosome is reduced to half, and that half number of chromosome is 23. Okay, half number of chromosome, which is 23, is known as the haploid number of chromosome. Okay, the original number of chromosome, which is 46, is known as diploid number of chromosome. Diploid, di means what? Two. That means half and half are combined together to give the diploid number of chromosome. Okay, this implies that during this stage of cell divisions there is reduction in the number of chromosome. Okay, which later at the end of the divisions will combined. That is during fertilization, when the male sex cells and the female sex cells fuse together in the fallopian tube. During fertilization the diploid number of chromosome is later achieved. Okay, in human, for example, the 46 chromosomes are reduced to 23 during gamete formation, that is spermatogenesis or oogenesis. Then as a result, each sperm or egg contains 23 chromosomes. The fusion of the sperm cells and egg cells, which I called fertilization that takes place at the upper part of the fallopian tube will now give us 46 number of chromosomes, which every human being has. Okay, the next we're going to look at now is the stages of cell divisions, which I've mentioned at this side of the board. Okay, I've mentioned this earlier, but in this video, we are only going to talk on the prophase stage. Okay, we have three stages of prophase in meiosis, and these stages are: we have the early prophase, we have the mid prophase, and we have the late prophase. These are the three phases or three stages of prophase stage of cell divisions. Okay, but because we don't have much space here on the whiteboard, we are only going to talk on the early prophase and the mid prophase in this video. Okay, if you look at this diagram here, this whole diagram have explained all that happens at the early prophase of cell divisions. Okay, here the nuclear membrane start to disintegrate a bit to some small degree. Then this is typical diagram of a cell. We have the central. The central here in the cell here we split into two and start to migrate towards the opposite side of the cell to form north pole and south poles. Okay, then after that, the next that occurs is the formation of chromosome. The chromosomes are formed by this structure here in the cell. This is known as chromatin thread. Okay, the chromatin thread here, as we know, is located inside the nucleus of the cell. And here in this stage of cell division, the early prophase of meiosis, this chromatin thread here condenses together with the nucleoplasm. We have the nucleoplasm here inside the cells, condenses with the nucleus, as we have here on the whiteboard, to form chromosome. And this is where the DNA, the genetic materials of every cells are located. Okay, DNA is located inside the chromosome formed. Okay, as I said before, listen, the chromatin thread here condenses to form chromosome, which is now seen here as a long thread. Okay, then here in the main prophase, this is the last phase we are going to talk in this video. Okay, at this stage of cell divisions, as we have on the whiteboard, the chromosome form will become shorter, thicker, and more visible. Okay, that is to become more clearly seen. Okay, it will condense, it will become more shorter, and here we have it as pair of homologous chromosome. Okay, then after that, the centrole will start to produce spindle fibers on which the chromosome will later attaches by their centromere at the next stage of the cell divisions. Okay, please let me summarize this again. Okay, at this stage, this is the early prophase of cell divisions, as we have here on the whiteboard. This is the early prophase. Okay, the central split into two to form north poles and south poles, and this here is the aster that help to hold the spindle fibers in place. Okay, by positioning the central. Then the chromatin thread will condense to form chromosome, which is seen as a long thread. This is what happens at the early prophase. Then here in the mid prophase, that chromosome which was seen as a long thread in the early prophase will become shorter, thicker, and more clearly seen. And of course, we have the centromere that hold this chromosome in place. Then we have pair of homologous chromosome. This because of the formation, how they are now arranged, because they have become more shorter, clearly seen, and more stronger. Then this central here will start to produce spindle fibers. Okay, this is all we have for now. Please, in the next video, we are going to talk about the late prophase before we move over to the next stage of the cell divisions. Thank you and God bless you. Please don't forget to subscribe to our channel.